Scientists are exploring ways to reprogram cancer cells back into normal, healthy cells. This concept is still in its early stages, but it represents a new and potentially transformative approach to cancer treatment. Here's a high-level overview of the topic. 1. Understanding cancer cells. Cancer cells are different from normal, healthy cells in several key ways. Uncontrolled growth. Cancer cells grow and divide uncontrollably, bypassing the regulatory mechanisms that normally keep cell growth in check. Genetic mutations. Cancer is typically driven by mutations in the DNA of cells. These mutations can accumulate over time and cause the cells to lose their normal function and become malignant. Ability to spread. Cancer cells can invade other tissues and spread to different parts of the body, a process known as metastasis. Cancer therapies, such as chemotherapy, radiation, and surgery, typically focus on targeting and killing these abnormal cells. However, these methods can be harmful to healthy tissues as well, and they often fail to address the underlying genetic and biological causes of cancer. 2. The concept of reprogramming cancer cells. The idea of turning cancer cells back into normal cells comes from the broader concept of cellular reprogramming, the ability to take a differentiated cell such as a skin or blood cell and revert it back to a more primitive, stem cell-like state. From there, the cell could theoretically be directed to develop into a healthy, functioning cell again. In recent years, scientists have made significant advances in this field, especially with the discovery of induced pluripotent stem cells (iPSCs). These are cells that have been reprogrammed to return to an undifferentiated state, similar to that of embryonic stem cells, allowing them to develop into any type of cell in the body. The idea is that cancer cells, like normal cells, may be susceptible to this type of reprogramming. 3. The process of reprogramming cancer cells. Process of reprogramming cancer cells to become normal again is still being refined, but it generally involves reactivating tumor suppressors. One potential approach is to reactivate genes that act as tumor suppressors. These genes, such as p53, normally prevent cells from becoming cancerous by repairing DNA damage or inducing cell death in damaged cells. By reactivating these pathways, cancer cells might be nudged back toward normality. Epigenetic reprogramming. Cancer cells often undergo epigenetic changes, alterations to the way their genes are expressed without changing the underlying DNA sequence. Researchers are exploring how epigenetic reprogramming could restore cancer cells to a healthier state. This could involve using chemicals or other treatments to alter the epigenetic marks that have gone awry in cancer cells. Gene editing. Advances in gene editing technologies, such as CRISPR-Cas9, have made it possible to specifically target and modify the genes within cancer cells. Scientists are exploring ways to use these tools to correct mutations that drive cancer and restore normal cellular functions. 4. Potential approaches and techniques. Several experimental techniques are being explored to achieve cellular reprogramming in cancer treatment. Gene therapy. Using viral vectors or other methods, scientists can introduce genes that may help reverse the cancerous state of a cell. This can include activating tumor suppressors, silencing oncogenic genes that promote cancer, or introducing genes that help guide the cell back to a normal state. Small molecule drugs. Researchers are also investigating small molecules or drugs that could target the molecular pathways involved in cancer reprogramming. These might help reverse the epigenetic modifications or activate genes that promote normal cell function. Cellular reprogramming factors. Some studies have explored the use of transcription factors, proteins that control gene expression that are involved in reprogramming normal cells into iPSCs. By introducing these factors into cancer cells, scientists may be able to push the cells back to a normal state. 5. Challenges and limitations. While the idea of reprogramming cancer cells is promising, it faces several challenges. Tumor heterogeneity. Not all cancer cells in a tumor are the same. Tumors can be highly heterogeneous, with cells that differ in genetic mutations, epigenetic changes, and behavior. This makes it difficult to find a one-size-fits-all approach to reprogramming. In complete reprogramming, even if cancer cells can be reprogrammed into a more normal state, there is a risk that they might not return fully to healthy cells. The process might lead to cells that are still capable of growing uncontrollably or developing resistance to treatments. Side effects. Reprogramming cancer cells involves manipulating the genetic and epigenetic machinery of the cells, which could have unintended side effects. For example, reprogramming may inadvertently induce other mutations or result in the production of abnormal proteins. Tumor microenvironment, the surrounding tissue and immune system also play a critical role in cancer development. Reprogramming cancer cells in isolation may not be enough to overcome the influence of the tumor microenvironment. 6. Current research and clinical trials. As of now, the concept of reprogramming cancer cells is still in the experimental stages. Some research has been done in the lab, using cultured cells and animal models to test different reprogramming techniques. For example, studies have shown that reprogramming certain cancer cell types such as those from the skin or blood into a more normal state can reduce their ability to grow uncontrollably. There have also been promising advances in gene editing and epigenetic therapies that could potentially be used to reprogram cancer cells. However, no clinical trials have yet been established to test this approach in humans. 7. Implications for cancer treatment. If successful, this approach could have profound implications for cancer treatment. Personalized medicine by reprogramming cancer cells. Treatments could be tailored to the genetic and epigenetic makeup of each patient's tumor, potentially leading to more effective and less toxic therapies. Reducing side effects. Traditional cancer treatments, like chemotherapy and radiation, often cause significant damage to healthy cells. Reprogramming cancer cells to revert to normal may allow for therapies that are less harmful to the body. Targeting the root cause. Reprogramming cancer cells could address the underlying genetic and molecular causes of cancer, potentially leading to more permanent solutions to the disease. Looking to the future, a field of cellular reprogramming is advancing rapidly, and while it may be some time before it becomes a mainstream cancer treatment, the potential is enormous. Researchers are working to refine these techniques and overcome the challenges associated with tumor heterogeneity, incomplete reprogramming, and potential side effects. In the future, we might see therapies that can transform cancer cells back into normal cells, offering a new, potentially more effective way to treat the disease. Until then, the quest to understand the reprogramming of cancer cells continues to push the boundaries of science, bringing us closer to a time when cancer could be treated at its core.